Over the last few lectures, we've been looking very closely at the structure of our proteins. Right? First, we looked at the structure of our individual amino acids that make up our primary structure. Then we saw how the interactions between our peptide bonds with the carbonyl hydrogen bond acceptor and the NH hydrogen bond donor could lead to various secondary structures. Finally, we looked at how the side chains could also interact between our different secondary structures in order to create these larger 3D tertiary structures. Now, of course, the last level of protein structure that we haven't looked at is quaternary structure. Now, quaternary structure is a little bit different from the other layers that we've looked at before, because it's still using essentially the same interactions that we saw with our tertiary structure, but now those interactions are occurring here between different protein subunits, right? So we have one protein here in the blue interacting with another protein in the gray, and those are bound together into a single functional unit. So essentially, the only real difference between quaternary structure versus tertiary structure is that it's occurring between unique proteins. So here we see the quaternary structure within a functional unit of the protein hemoglobin. So our hemoglobin has 4 individual polypeptides, 4 individual protein units. You can see those marked with the light gray and the darker gray, and the light blue and the darker blue. But again, these proteins don't function individually. They only function when they're all bound together in this larger complex. So this is our quaternary structure. We would refer to this as a multimer or an oligomer, right? Because we have multiple units combined together to create this larger structure. Now, there are a wide variety of different ways that we can combine together our proteins to create different oligomers or multimers. So here we have a protein represented by this blue ball here. If we combine 2 proteins together, we now have our quaternary structure that's an oligomer of that specific protein. Now we can be a little bit more specific here, and we can name this quaternary structure based on the number of individual protein monomers here. So we would refer to this as a dimer, since we have 2 different protein units together in this oligomer. If we then add on 2 more, this would be a tetramer here. And of course, we can keep adding those on, and we just use a different prefix to indicate the number of units that we have added together here. So this would now be a hexamer with 3 of our dimers combined together into this hexagon shape. Now, again, depending on the protein, this can continue many times to create very large oligomers here, right? So in this case, we are taking multiple hexamers and we're stacking them together to create kind of this fibrous-like structure. The yellow shown here is just to show the individual rings of our hexamers as we're creating this larger structure. Now, in addition to naming how many of our units we have in these different oligomers, we can also name our oligomers based on what kinds of proteins they have. So specifically here we have a Homo Dimer or Homo oligomer. So the Homo Dimer, of course, would be this case right here where we have a dimer of 2 of the same molecules or 2 of the same protein molecules. And our Homo oligomer would be over here with our large structure with multiple units. Now here, of course, we see a very uniform structure forming as we're adding these different protein subunits together. But this is not always the case for our homodimers and homo oligomers. So this protein shown here is a bar domain protein. So they're used to help create some of the interesting structures that we need as we are doing endocytosis in our cells and pulling in new materials from the outside of our cell. So an individual bar domain protein is marked here in purple and in green. And this is actually a homodimer, right? These are identical units, but they're not linked in the same kind of regular way that we saw in the previous slide. So here we see our linking site between our 2 proteins is over here at the end of our protein. And so we get a slightly different shape here as we link these together. So depending on what protein you have and kind of where those interactions take place in order to link those proteins together, you can create a variety of different shapes and structures within your quaternary structure. Okay, so far we've only looked at homodimers, right? Where we're taking different units of the same protein and then combining them together in interesting ways. If we're combining two unique proteins, we're forming what we call a heterodimer or a heterooligomer. So within our antibodies, you have that typical Y shape, which is actually formed from two different heterodimers combining together. Right, so you might remember the term of the heavy chain versus the light chain of an antibody if you've studied antibody structure at all in any of your biology courses. So we have this light red heavy chain here and the darker red heavy chain here. So those are our two heavy chain units shown in our little cartoon down here. And then we have our lighter chain protein marked in blue that's bound to each of those heavy chains. So we have a dimer of dimers, essentially, where we've got our two red heavy chains dimerized together, and then those are also dimerized to our light chains. And again, notice you can create some very interesting structures using this sort of oligomerization. Now, the interactions for these, again, are very similar to what we see for tertiary structure. So you can see we have these interchain disulfide bonds pointed out specifically here. These would help to link these chains together in that semi-permanent manner, at least as long as you don't encounter any reducing conditions. So we can see disulfide bonds, we could see electrostatic interactions, we could see hydrophobic interactions, any of the usual interactions that we see with tertiary structure. It's just that now they're occurring between different protein subunits. Now, if we want to measure our protein oligomers, we can use a lot of the same approaches that we've looked at for looking at individual proteins. So here we show a analysis of a set of oligomers using protein chromatography. So this is size exclusion chromatography. We're reading our proteins that come off of our column using the absorbance. So we'll get a higher absorbance when we have more proteins coming off. Now, at size exclusion columns, the largest proteins pass through first, because remember, they're excluded from those small pores that the other smaller proteins will travel through. So they pass through first. So we're always going to see our largest oligomers coming out first. So in this case, we have a decamer as our largest oligomer with an octamer coming out shortly after. Now, of course, all of our other oligomers will come with at repeated intervals after our initial oligomers. So in this case, we have hexamers, tetramers, and trimers, and then a dimer and a monomer. Based on the area of these peaks, you could actually then calculate what the relative composition of your sample is, right? You can compare how much of your protein is found in the octamer versus how much of your protein is found in the monomer. And again, it's quite common to see proteins oligomerizing this way. So it's not rare to find different peaks here kind of showing different oligomers that might tend to form. Now, our size exclusion chromatography is one way to separate and identify proteins by their molecular weight. But of course, SDS page electrophoresis is also a very good way to analyze our proteins. So again, our proteins will separate based on size, as they always do in SDS page. But of course, in this case, we're looking at the same protein, but with different oligomers. Right, so we have a big strong band here of our monomer. So our monomer is right around 66 kilodaltons in terms of its molecular weight. And then we see distinct repeating bands based on the molecular weight of our monomer. So if we double the molecular weight of our monomer here, we get the weight of our dimer, which is 132 here. And of course, this pattern would repeat, right? We could triple it to get the mass of our monomer here. and so on and so forth, right? We'll get a very characteristic repeating pattern based on the size of our monomer. Now, if you had a heteroligomer, this would get a lot more complicated because then, if you measured the molecular weight of each of your individual monomers, you could say, okay, a heterodimer is pretty easy, but then for a heterotrimer or a heterotetramer, you have to know what the ratio of your different units is within that larger oligomer. So this is easiest to use when you're analyzing different homoligomers. It can be used for heteroligomers. So with this quick look at quaternary structure, we've seen some of the different types of quaternary structure with homoligomers and heteroligomers. We've also seen how they're bound together, right? Again, using the same kinds of interactions that we see with our tertiary structure. Now, I just want to emphasize here that this sorts of quaternary structure is actually incredibly common and incredibly important within the body. So many of our most important functions are performed by protein complexes where we're combining different proteins together in order to carry out that specific function. So on the left here, we have the ATP synthase, which synthesizes all the ATP that we use in our bodies to, again, carry out most of our different activities. We have the ribosome here in the middle, which is a complex, of course, that we use to synthesize proteins. And on the right, we have a DNA replication complex, which is involved in, of course, replicating our DNA to make sure that we keep our genetic information intact. So really, every area of what our body does depends on protein complexes. And again, these protein complexes really emphasize the complexity within our body, right? We really can't just identify one protein that might be critical for a specific thing. Often we have multiple proteins that are working together to accomplish something. So it becomes even more difficult to identify what the key components are and kind of what each individual part is doing in order to make that complex function. So I hope this kind of shows you a little bit of why our body is so complex and it is so difficult to figure out all of the unique biochemistry within our cells.